Hello, my name is Ander Diaz. I am a postdoctoral fellow at the University of Toronto and the Ontario Institute for Cancer Research, and I'm going to present my project entitled In Silico Generation of Synthetic Cancer Genomes using Deep Learning Algorithms. First of all, I would like to show you why we think this is an important project. During the last few years, several international consortia, including the Cancer Genome Atlas, the International Cancer Genome Consortium, and the Pan-Cancer Analysis of Whole Genomes, and more recently, the Heart with Medical Foundation, the 100,000 Genome Program, and the ARGO Consortium have been collecting sequencing data from numerous patients in order to study the genomic characteristics of different tumor types. However, this data contain confidential patient information and therefore access is restricted. In addition, although the tumors present in these repositories have been analyzed in depth, their ground truth is unknown, so it's difficult to use this data to develop new algorithms. Knowing the ground truth is very important for training and benchmarking cancer analysis pipelines. In this sense, some strategies that have been used are, for example, the manual review of several mutations to obtain a high-quality curated database, the use of tools that generate in silico mutations, or using gold standards. However, manual revision is very time-consuming and does not guarantee that there are no false positives or negatives. Actual tools that add in silico mutations generate a lot of unwanted artifacts and reflect simplified and limited mutational models. On the other side, we don't have enough gold standards. Moreover, they were sequenced with older technologies. Because of that, in this project, we propose to use generative models, such as generative adversarial networks and tabular variational autoencoders, to simulate high-quality synthetic cancer genomes that can be used to design new and better programs to perform more accurate genomic analysis. Here, we can see an overview of the methodology that we are using to develop this pipeline. First, we are taking 8 different tumor types from the PICO analysis. Then, to improve the simulation, we process the data and break it down into individual features. Depending on the nature of the characteristics, we use different approaches and models. For example, for donor and driver characteristics, we train different generative adversarial networks models. On the other hand, to simulate the genomic position of the mutations, we use tabular variational autoencoders, since it works better to incorporate the long context relationships. Finally, since the variant allele frequency is related to sample purity, we randomly select frequencies according to the real distribution in patients. For example, if a simulated donor has a low variant allele frequency, they will have more mutations with lower frequencies as well. To be able to say that our synthetic genome simulations are good, we need to compare them against the real data. Therefore, first we saw the number of different mutation types each patient has, from single nucleotide polymorphisms, dinucleotides, and trinucleotides, to small insertions and deletions. In this plot, donors are represented as dot, in orange real ones, and in black the simulated. So in these preliminary results, you can see how the heterogeneity of each type of mutation is well simulated for this specific cancer type. Second, we need to focus on where the mutations are simulated. Although they occur randomly in the genome, each tumor type has a distinct pattern. For example, patients with chronic lymphocytic leukemia have a large concentration of mutations in the immunoglobulin genes. When we represent the position of the mutations for the simulation of this type of tumor, we can also see how these characteristics are conserved, demonstrating the ability to generate tissue type-specific mutation patterns. This is also confirmed with an overall Pearson correlation of 0.82. The third point is the frequency at which each mutation is found. This is important in the sense that it allows us to simulate different clones and samples purity within the same tumor, just as it exists in real tumor genomes. This is only the example for one tumor type, but you can see how the simulation is very similar in both the overall variant allele frequency and the mean variant allele frequency per donor. Finally, we verified that we can detect the same mutational signatures present in patients. In each tumor type, the biological mechanisms that cause the mutations are different, and these can be detected as different signatures. Some of them are shared among almost all tumor types, such as the signature associated with aging, but others are specific to a tumor type, such as the mutations generated by smoking in lung cancer. Being able to simulate the specific signatures for each tumor type is important to achieve very realistic synthetic genomes. In these plots, you can see how the mutational signatures detected in the synthetic genomes are the same that are present in the real tumors, with the same density of mutations and the same proportion. We can therefore conclude that the model strains so far resemble real tumors. To confirm that, we decided to use Deep Tumor, a machine learning algorithm that predicts the tumor type of origin using the list of somatic mutations. In this case, it is important to mention that CLL has two main subgroups, depending on the mutational state of the immunoglobulin genes, mutated CLL and unmutated CLL. When distinguished between subgroups, Deep Tumor accurately classifies 91 and 98% of the unmutant CLLs for real or simulated donors, respectively. However, mutant CLLs is very similar to lymph BNHL tumor type, being those more difficult to predict. This also happens for our simulations, suggesting that we are also simulating the CLL diversity. The conclusion is that our pipelines allow us to simulate very realistic synthetic tumor genomes. By providing a freely distributable set of realistic cancer genomes with non-growth truth, this tool enables the benchmarking of cancer analysis algorithms and the improvement of existing programs. Thank you very much for your time.